All right, so the cyber infrastructure project that I'm going to talk about is one component of another larger project that we had with involving undergraduate science teaching, and it was funded by the National Science Foundation in 2004. So that project, known as CASP, CASP stands for the Center for Authentic Science Practice in Education. And the idea of this project is that we are integrating science research into the undergraduate teaching laboratories in the first and second year. So that actually brings together several components. First of all, undergraduate research is one of those components of science teaching that the literature demonstrates helps us retain students and, of course, helps us prepare them better for the technical disciplines. But bringing it into the curriculum was one of our first challenges as a way of trying to get students engaged in undergraduate research in a way that didn't require them to be the ones doing everything extracurricularly and going and seeking out opportunities in addition to what they were already doing, so that those students who maybe hadn't considered the possibility of research, we can reach them in their classes. But not only are we bringing it into the courses, that's one challenge, but we're bringing it into the courses in the first and second year. So very, very early on, most students who do undergraduate research in the sciences are doing it in their junior or senior years. And so this project helped us create the model that would allow us to engage students in undergraduate research, real undergraduate research, as part of their coursework in these early years. So to achieve that, we had three components to our strategy, what we call the modules, the instrument network, and peer-led team learning. So the modules are really the heart of the content part of this, which is where we worked with scientists at Purdue and elsewhere to basically translate their research work into activities that undergraduates could do in a period of about half of a semester in these undergraduate science laboratories. When scientists think about doing undergraduate research with undergraduates in their laboratories, they're usually thinking of sort of a summer project where a student comes in and they're an apprentice in their laboratory for a few weeks. But if you do the math, a few weeks, say 8 weeks in the summer, working, if it's half-time, 20 hours, but if it's full-time, 40 hours a week, that's quite a bit more than the half of a semester, which is about 8 weeks, one lab period a week, which is 3 hours. So you've got maybe 24 hours of actual contact time with these students when they're actually doing research. So hence the challenge of this is, how do you engage the students in something that's actually meaningful research, not just a meaningful learning experience for the student, but actually going to be returning meaningful data to the scientist in a way that this really engages the scientist and the student in a research relationship. How do you do that in a 24-hour contact period? So our modules are the heart of actually doing that, where we take the research work and we decide which activities are appropriate for the students to do in the laboratory and actually end up giving data or samples back to the scientist that are usable. But an important support part of that is the instrumentation network, which is what I'm going to focus the rest of this talk on, which is the cyber infrastructure part of it. Because if you're going to have undergraduate students, especially first and second year, doing research where the data are usable by the scientist, then they can't be collecting this research data on the kinds of instruments that are typically found in a first-year undergraduate laboratory course. While those instruments are perfectly good for the learning part of it, if the research data are going to be ultimately publishable, likely the students are going to need to have access to research-level instruments. How do you do that and still maintain sort of the scope that we wanted to hit, i.e. many hundreds of students getting these research experiences? You can't buy a bunch of these instruments and put them in every individual lab. So the answer is you buy one of the instruments and you make it accessible via some remote method, which is the key to this. We have to maintain security. We have to maintain ways that the students can't just get on there and basically ruin the instrument. I'll get into the details of that in a bit. The last bit here, just for completeness, is what we call peer-led team learning. This is a form of supplemental instruction where students are engaged with each other. The more experienced students are helping guide the least experienced students through the process. And this, of course, parallels what happens in a traditional research group in a science laboratory. So the remote network, as I said, the idea is that we basically purchase one of each of the types of instruments. We equip them with auto samplers, we enable remote access, and then all of the groups who are involved in this can use it. One of the things I didn't mention is that the CASP project is not just at Purdue. Purdue is the heart of it, but we have 17 institutions across the nation and one international institution that are involved with us in this. And so when we say remote access, we really mean remote. So this is a photo of the sample holder area of our Raman spectrometer, and it just shows the auto sampler in this case is a small 16 sample remote, or rather auto sampler, where the samples are in, if you are familiar with it, standard NMR tubes essentially, and we can ship those very easily and then stick them in our auto sampler there. Okay, so the way that this is set up, basically I'll start over here. We've got an instrument, any of the instruments. This could be any of the instruments. So then the instrument has a server connected onto it, and it runs the instruments, physically connected to the instrument. And the software that runs that instrument is served to the outside world through the Citrix client. So the Citrix client that was made available to us through a collaboration with ITAP allows the student or any user to operate this instrument without actually having to have the software on their own computer. So they can basically work through the equivalent of software remote to use the software that runs this instrument. Because we can really only have one user at a time on the instrument, we wanted to make sure that students and users got onto the instrument and back off as quickly as possible so that the next student can then use the instrument to run their sample. So we didn't want students collecting data and then sitting there and processing the data or analyzing the data using this server. So as soon as the student collects the data here, the data are dumped onto a data file server, and they log off of the instrument, freeing up the instrument. They come onto here using the same approach, using the Citrix interface, and then they can process their data there. Now we can have as many users on here simultaneously as we want, so that students can be working up their data at any time, day or night, that they want to or need to. So our instrumentation right now consists of an FTIR Raman spectrometer, high-performance liquid chromatograph, gas chromatograph, and a gas chromatograph coupled with a mass spectrometer. We anticipate additional instruments for teaching, and a development that we're currently exploring for this model, even though we received the funding to do this for CASP, which is an education-focused approach, we're starting now conversations with a couple of the centers in Discovery Park to see if some of their instruments that may have high usage could benefit from also being connected through this model. So while this model was developed for this teaching approach, in fact, it has potential for any kind of a research-based approach where people need to access an instrument but not necessarily be sitting at that instrument the entire time that it's being used. or maybe somebody is collecting a sample remotely and wants to use the instrument that's located in one specific location. This is a way to get them to there. So there are a variety of ways that we have used the instruments that are connected through this system. And I'm talking now for the CASB approach for education. The standard batch mode, essentially the students submit their samples and we run the samples for them and then return the data files to them by putting those data files on the file server, and then the students access them there. And this is really used when an instructor has very beginning level students. They want to introduce the students to the use of data from these instruments, but don't necessarily feel that the students are ready to really get on there and start using the instrument themselves. The individual mode is where advanced students have received some sort of training to use the instruments, and this includes a combination of our tutorials that are online on the CASB website and instruction from their own teachers. And they can access the instrument and run their samples much like I described on the previous slide. Then they access and process their own data. Something in between is a demonstration mode where an instructor may choose to show a group of students in a lecture by actually logging onto the instrument, running something live. And I've actually done this on some occasions when I give a lecture about this. And then you can actually teach the students how to use the instrument, what the data look like, but not necessarily have them using the instrument themselves individually. Now, on all of our instruments, we have video cameras in the laboratory that allow for students to watch as the process is going on. And this really lets people know that this is a live instrument collecting a live sample real time, not a simulation or something that we did at some other time and we're just showing you the video or something along those lines. And those video cameras, for example, in some cases we've had the laboratory manager actually communicating with the user through the video camera so that there's really a live interaction there, not only with the instrument but with people who are operating the lab. We are able to modify the software that runs these instruments. So this would be the full version of the software interface, with all the menus available. But as I mentioned, we know that we can't necessarily give all that functionality to a beginning student, especially students who are not our students, and we really don't know what kind of ethics they might have, because of course our students would never do anything we'd worry about. But we can strip out a lot of the options and basically just leave the on and off button and the run button. And that makes it so that we're protecting not only the instrument but also in some cases the underlying file structure of the data that's there. As I mentioned, we have the video cameras. So this is an example of the interface you get for the video camera itself. On this menu here, this drop-down menu, there are some presets for the video camera, so you can zoom in on some part of this instrument as you're actually running things. So here's an example. You're running the sample, getting your data, and actually looking at the data interface part of this instrument. Now this instrument actually is not very interesting because it has to be closed when it's running. So the people who look at this instrument really are just looking at these 4 LEDs. A more interesting example is our gas chromatograph, where on this turntable here there are 96 positions for samples. And this little arm here is actually a robotic arm. It's got 4 little pincer grips right there. And so when you go to run this instrument, you can actually watch this rotate to the position of your sample. Here's a sample vial right there. Pick it up, drop it into the tray, and then you can actually watch as the injection takes place and your sample is being drawn into the system. And of course all of the LEDs over here. So you can position the camera using those presets that we have to actually watch as each of those parts is taking place. We have some more information about the project on our website, so if you'd be interested in learning more, you could go there. And with that, I will pass it back to Peter.
Okay, excellent. Well, thank you for that kind introduction. Thank you for the invitation to come and talk today. This is a great meeting, and I'm very pleased to see this happening at Purdue. I've been here 10 years now, and cyber infrastructure and the need for it has took a while for people to realize, and it's certainly starting to pick up, and I think with Peter's arrival, that's going to be significantly catalyzed. So what I would like to talk to you today in the next couple of minutes here is a little bit of my experiences at Purdue in terms of developing cyber infrastructure to help underpin a large-scale genome project that we've been running at Purdue for about the last 10 years. And what I'm going to do initially is actually very briefly introduce the science to you so that you get a feel for the type of infrastructure that we need to actually underpin that science, as opposed to kind of doing this in the other way around. But I'm not going to dwell too long on the science, but just to give you a flavor of the kind of things that we do so that you'll then be able to better understand the need for the cyber infrastructure that we developed, and then I'll go on to explain that cyber infrastructure just in a little bit of detail. This is obviously the introduction. These are the people that I've been working with over the years. The people in gray are now no longer part of the group, have moved on to bigger and better things, and the people in bold there are still part of the team. Is it a pointer up here? This button here? Oh, I've got this one. Okay, all right, technology. Okay, so let me first of all briefly describe what Ionomics is, because the Ionomics project is what this cyber infrastructure that I'm going to talk about supports. So basically, Ionomics is a project that we've been developing, me and my collaborators, to basically try to understand the genes and gene networks that underlie the regulation of the elemental composition of an organism, or its chemical composition. So when you eat your Frosties in the morning and you look on the back of the Frosties, whatever other cereal that you're interested in, you're going to see a whole list of vitamins and mineral nutrients that you require for your existence, for your basic biochemistry, including things like zinc and calcium and iron and manganese and magnesium. And there's a long list of these things, macro and micronutrients, which are essential for all living systems. I primarily work on plants, but these elements are also required for animals as well. And I list those elements there. You don't particularly need to pay too much attention to that. But there's basically a list of about 20 elements that we're very, very interested in, both essential micronutrients and macronutrients, but also potentially toxic trace elements like arsenic, cadmium, and lead. And the projects that we run in our lab are basically to measure these elements in high throughput using various different kinds of technologies, liquid handling robots, 96-well microtiter plates, an instrument called an inductively coupled plasma mass spectrometer, and we actually use all these and apply this measurement technique to various different systems. The primary one that we work on is Arabidopsis thaliana, and that's what I'm going to briefly touch on today, but we also have projects working with mice, working with yeast, and working with rice. And so I guess since we deployed the cyber infrastructure that I'm going to talk to you today about, which underpins this project, we've collected almost 4 million data points, so quantitative data points describing the concentration of these various elements in over 220,000 samples. And so right there you can see that clearly, having a notebook and a couple of Excel files is just not going to cut it in terms of handling that kind of volume of data. Okie dokie. I had the same problem when I gave the McCoy talk, and it was on this exact computer. Can we make this go forward, please? Yeah. These guys don't seem to want to do anything. No. Oh, there we go. Okay, so 2 quick examples of what we've been doing. One example that I'm showing here is actually we recently, well, it actually took us about a year to do this, is we took the full yeast genome deletion and overexpression collection. So that's about 5,000 strains in which each individual gene in yeast has been deleted, and about another 5,000 strains in which every gene has been overexpressed in yeast. And we took that full collection, represents about 12,000 different strains. Took us about a year to collect full ionomic data on all those strains. With replicates, it represented over 51,000 samples. And I'm just showing you a nice little kind of heat plot of some of that data. So as you can imagine, this is a tremendous data resource that can be mined by many, many researchers to start to try to understand gene function in yeast, but without having cyber infrastructure to underpin both the collection of that data and the management of its dissemination, this whole resource becomes really next to useless. Another nice example of the kind of things that we can do, or a nice example of the kind of things we can do with this yeast database, just to give you a quick feel, is what we did is we took the data and we actually looked for clusters, looked for ionomic profiles of the data which form particular clusters, and we found this interesting cluster here which represents genes disrupted in mitochondrial function. And actually when we went ahead and we overlaid onto that genes of unknown function, or genes that have had no function annotated, we could identify a whole new slew of genes which are obviously involved in mitochondrial function because they fall into this same cluster. So that's just one example, again, of what you can see with this kind of data, but you obviously need a cyber infrastructure to underpin that. And then one last quick example, this is one from Arabidopsis thaliana. This is a project where we took 360 different populations of Arabidopsis thaliana, which have grown in different parts of Europe, and which have presumably become locally adapted to the particular condition that is prevalent where they exist. And by analyzing the elemental composition of these 360 Arabidopsis thaliana populations, and then connecting that phenotype to the changes in the genome using a technique called genome-wide association mapping, which I'm not going to go into, we can very rapidly get to genes that regulate the elemental composition of these plants, which is important for multiple reasons. But again, you can see how the scale of these experiments, 2,200 samples, almost 50,000 data elements. Again, without an enabling cyber infrastructure, you're not going to be able to make head and tail of this kind of data. Okay, so how did we go about building the cyber infrastructure which has been required to underpin this type of research? And I guess this really goes back about 10 years. I'm not going to give you the whole history. But very briefly, and I think this is kind of maybe symbolic of a lot of these kinds of projects, initially the cyber infrastructure was very, very primitive. It was essentially Excel files, basically curated by the biologist doing the research. And very quickly you realize that Excel files are not a good strategy for many, many reasons, which I'm sure you're all aware of here, not the least being that it's impossible to search across all Excel files for a given data element. So from Excel, we then were able to transition to Access, Microsoft Access, which required funding or convincing an engineering student to come to my lab for one summer. So we transitioned our data into Access, but that still really wasn't even close to optimal. And particularly because what we wanted to be able to do was to web enable this system so that actually our collaborators could also send data to us, and perhaps more importantly, we could actually disseminate the data that we were collecting to the public so we could broaden the base of people who are actually interested in analyzing this kinds of data. And so that's when we got involved with what used to be called the E-Enterprise Center, and I think the E-Enterprise Center then evolved into the Cyber Center, and over the last 4 or 5 years, our system has really been transitioning from Access to what it is today, which is basically based on Postgres and Tomcat, and very soon be transitioning to virtual servers, and it has a very nice sophisticated front end and a very powerful database back end. So that's kind of the brief history of how this project worked. It was all basically funded from NSF and NIH grants, and it was quite a struggle to get this thing going, and I'm going to make a plea right now, and that I think it's very, very important that Purdue pays attention to the fact that, faculty members, it's very hard for faculty members to get the resources to develop this type of infrastructure, which I would strongly argue is research infrastructure in the same way that the university provides teaching infrastructure. They need to be providing research infrastructure as well, and don't expect faculty to both build it and deploy it. Okay, so to continue, what does this infrastructure look like? So there's basically a back end and a front end. The back end is the database and what I would call the system which we use to manage the workflow of the projects. I don't want to go into too much detail, but essentially we have a system which breaks out the workflow of our project into particular modules, particular activities, and when the researcher is doing that particular activity, windows open up, the researcher can put into the system the information or metadata that describes the experiment. And as the researcher goes through the experiment, these different portals, windows open up in this web-based system, allowing the researcher to capture the critical pieces of metadata. This is based on a control vocabulary, so we've all decided to agree that particular words mean particular things, and we're working towards transitioning this to be more ontologically based, so you could actually do interrelationships between terms. Continue on, you collect the data. We have portals for letting the operator of the instrument not only collect the metadata about the instrument, what the settings were, etc., but also then to upload the data from the instrument via the web onto the system. That's nice because it lets us actually collaborate with people. I have a collaborator in the University of Nevada in Reno, for example. I have a collaborator at Dartmouth. Those people can run samples in their labs and upload the data to the same system. We also have tools on the system which let the instrument technician, the person who understands how the data was collected, lets them polish the data before the data is then pushed and released to the public side of the database, which is then accessible to both the public and to the people in my lab and in other labs. And it's been designed in such a way that actually the database has both a private and a public side. So if you have a collaborator you're working with samples that don't particularly want to go live to the whole world, then you can keep that information private. We do this because NSF is obviously strongly involved in funding this project and is strongly interested in disseminating data very broadly, but obviously you've got to balance that with collaborators and your own research group, in which you don't want to immediately release the raw data, so you need to have that kind of balance. Once the data's been pushed to the database, you then have tools that let you search the database. You can search on multiple different parameters. I don't need to go into all the details, but you search the database, it pulls up experiments that may be of interest for you. Once you get those experiments, you see summary data of the experiments, and then there's various tools that you can use to actually look at the data, different kinds of plots that I'm showing here. You can also download the data to your own instrument, your own computer, to do whatever kinds of analyses you want. Approximately 95% of all the data in our database is actually publicly available. So anyone can download it and manipulate it and work with it and do what they would like. We also have a picture of every single experiment in the database. It's obviously a Arabidopsis thaliana, so people can see what the critter looks like. This obviously could be a mouse or whatever you're interested in. So that's the back end. We collect the data in a control workflow, control vocabulary. The data is all stored in a relational database. Not only is the raw data there, but also the metadata, which explains and describes the data. Very, very critical if you want to share and interoperate the data with other labs so they understand what your data means. On the front end, we have what we're calling the Ionomics Hub. This is the other hub. This is the hub that the hub guys don't mention. Because it doesn't use the hub technology. We're based on a different kind of approach. But it's another hub. It's a generic hub. We call it the Ionomics Hub, funded from the National Science Foundation. And this is kind of being developed as a community portal, not only to let people access the data, so you can click the various databases here, which gets you into the data, which has been collected through that backend, which I described. So this is a Arabidopsis thaliana database. It's a rice database. And that's the yeast database, which contains the yeast data that I also described. There's some search tools here. There's some summary statistics about what's going on in the database. There's a little bit of information about what's in the database. And there's a link to a Wikipedia article, which describes ionomics and is updated quite regularly. Then we have a few little tidbits of information here to build the community. And this whole thing acts as a way to disseminate our data. So it's getting a few hits, nothing like the Nano Hub, but we build up a dedicated following. We're up to close to just over 5,800 unique users of the database now who come and regularly look and view and download the data, pretty broadly distributed around the world. So it functions both to disseminate our data and also performs a very good outreach role. So that's what we've got right now. So what are we developing? So we have several projects that we're developing. So this project here is called the Arabidopsis Ionomic Atlas, which is kindly supported from the seed grant from the Cyber Center. So this is the next step in the project, in the sense that what we now want to do is not only to have the data that we've collected in the lab, but now to understand how the genetic variation, which is altering the elemental composition of organisms, is actually involved in controlling the geological and geographical distribution of those organisms across the globe. And that's going to help us understand how plants are modifying and adapting to global climate change, for example. And so this is a prototype of that system that we're developing. It's still in the early days yet, but basically what it does, it lets you map populations of plants. It lets you then overlay the genotypes of those plants and then environmental factors that may be affecting the distribution of those plants. So what else is coming up? So we also have just recently been funded from the National Science Foundation to further enhance and develop the Ionomics Hub. We've really got a pretty good handle on the data collection side, the back end. What we want to do now is to develop tools which will start to extend and enhance the community of users of the database, and also to spread that community to start adding not only more primary data, but also to start adding more analysis, more knowledge into the database. So we need people to come to the database and start to annotate the information and add knowledge. So we're going to develop a whole bunch of new and interesting tools, which is going to allow the community to not only share data, to annotate data, to add new data, to develop work groups. We're also going to roll out very, very soon a new tool which we call the Lit Cloud, or the Literature Cloud. And the idea behind the Literature Cloud is, as you probably all know in your own fields, many, many thousands of articles are coming out in your particular area every month, and it's almost impossible to follow everything. So what we're trying to do is basically kind of crowdsource out article filtering. So basically take a whole bunch of individuals who are interested in filtering their own articles and actually have that information fed to the iHub, and then presented to the users of the iHub as a way to try to filter out literature and get to those papers that you're particularly interested in. And that was another project that was supported by the Cyber Center, and that will be rolled out on the iHub probably in the next couple of weeks. Okay, so what's been the impact of all this 10 years worth of work, building the cyber infrastructure? So I can say since 2007, when we actually, what I would say, rolled out our probably first really mature version of the Ionomics Hub, my group has published 20 papers which would have been impossible to publish without this infrastructure. My group has been able to get awarded over $10 million in funding, which I could make a strong case that NSF and NIH would not have been interested in funding unless we had this infrastructure in place to show that not only could we do this large-scale functional genomics project, but we could also disseminate that data almost instantaneously to the general public. Two things which NSF and NIH are very, very keen on. And then I think maybe one of the things I'm most pleased about, and this is slowly getting more and more, but now there are 5 papers which are out in the literature, and these papers are basically not solely, but to a large degree based on data that's been accrued from our database. Nothing to do with our group. We don't ask permission for people. Our data's public. People have gone in, they've taken data from our database and built their own papers out of that data, including papers in top journals like Plant Cell. So I'm very, very pleased to see that. So with that short introduction to Ionomics and rapid flyby of cyber infrastructure that supports Ionomics, I hope I gave you a bit of a brief flavor of what we've been doing. And if you have any questions, I will certainly try to answer them for you.
Hi, everyone. Sorry about that little delay. My name is Matthew Potrawski. I'm the Managing Director of Prism, which is the Center for Prediction and all the rest of the stuff that Pete Baker just announced. We're an example of a project here at Purdue that is using cyber infrastructure to accomplish the goals of our program and our mission. So that's what we're going to focus on today. Overview of the program. The program is led by Dr. Jayathi Murthy. If you remember in John Campbell's presentation, he was noting a couple different hubs. Our program does have a hub, although most of the cyber infrastructure that we utilize is not run through the hub. It's through the community cluster aspect of what John was mentioning. We are a DOE funded, specifically under the National Nuclear Security Administration program. We're just over $20 million for a 5-year program. The goal, we are involved with 3 other partners, UIUC, University of Illinois, University of New Mexico, and Vanderbilt, just recently this year. Our program involves 35, 40 different faculty, students, grad students, and postdocs across all our partners and in Purdue. Our focus is on the petascale and computing, and we'll talk about that a little bit more specifically, why it's that. Specifically, the device that we are developing and testing for the predictability and reliability of that device contains a lot of really stringent requirements for an NSA. So we'll talk about that a little bit more. Another component of our program that doesn't involve the cyber infrastructure but is equally important to the PRISM program is the experimental aspect of it. Because as though we develop this device and simulate its reliability within the cyber infrastructure, we also are doing some verification and validation of that device experimentally as well. Our mission is to substantially accelerate the development of those MEMS technologies. MEMS is a micro electromechanical systems, which basically are real small devices. We are trying to significantly improve the understanding of that long-term reliability, specifically because of the requirements by the NNSA, which are, we'll go into a little bit more detail, are very harsh requirements and large cycle times. So we have to cycle the usage of the device a lot of times, and that's why we can't do that manually. So that's why we're using simulation to accomplish some of these. Another aspect, a real key component aspect of what this program is the incorporation of UQ, which is the uncertainty quantification. Whenever we talk about the development of models and simulations, as academics, we like to say, this is what we've done, and here are our assumptions, and be very specific about what we are assuming. But we all know that those assumptions, there's some variability in those assumptions, which is uncertainty. So the key component that the NNSA wanted us to include in this program is the incorporation of that uncertainty into the simulation model as well. Before I brought this slide up, I was going to ask, but I'm trying to get through here. How many people in here think that they have a MEMS device on them right now? Basically, everybody's hand should be up. If you have a cell phone, a laptop, or anything, all of these types of devices are now having MEMS devices included into it. If you have an iPhone, the accelerometer that allows you to change it, that's a MEMS device. If you have a laptop or a cell phone, those that have microchips in them. So MEMS devices are becoming very, very prevalent. So there's a limit to what the NNSA or the government and defense agencies will allow them to be included into those devices. So that's kind of the impetus for this program. We talked about the performance requirements. I want to get a little bit more detail. These are some really stringent requirements. It must survive billions of cycles. And sitting there flicking a switch a billion times is not something that's going to happen in any reliable amount of time manually, or even mechanically from another device. So that's a big component of why we're using simulation. Very dynamic impacting conditions. These are not pristine environments where these devices are going to be utilized. So we can't just say, okay, here's a nice little shelf that it's on, it has to be able to work there. It's going to be in harsh environments. High G, for example, up to 30,000 G for over short periods of time, over milliseconds. Those are some pretty harsh conditions that these devices have to sustain. Large range of operating temperatures from -50 to 80 degrees Celsius. That's pretty cold and pretty warm as well. So these types of conditions are not something that are easily experimentally tested. So thus the need for simulation. The key component, one of the things that we're doing about is focusing on the types of failure mechanisms that we're going to be testing. Once again, another component of why simulation is important. These types of failure mechanisms can range anywhere from class 1. No moving parts, nothing happening. The device is a very static device. All the way up to class 4, which has moving parts, areas of impact, areas where the components of the device are rubbing together. So there's components of wear. This is the area of focus that Prism is working on in the device that it's testing. So the types of failures that you experience on these types of devices are anywhere from friction to wear to stiction, those types of things. So this is the area that we're focusing on these devices. Here's a graphic of the device that we're using we've defined to use for our research. It's a three-dimensional one, and I'm going to show you another picture in a few minutes that make an image that you're looking at the device from this direction. So this front end right here is what you'll see in the next graphic about that. But this device is a very simple capacitive RF MEMS switch. It's built up of a dielectric, a membrane, which is actually the part that is pushed and actuated, the anchor for the switch, and then an electrode. So this is the device that we are utilizing as the test device to do our research. Failure mechanisms. The types that we are really focusing on cross over a lot of different areas, not just simple wear and tear. Talking about dielectric charging, contact area damage, current channeling, mechanical failure. I'm kind of flying through this kind of quick because we're a little bit short on time, so we can address a little bit more questions in question and answer time. But these mechanisms are poorly understood at this scale. When we're talking microelectrical in the MEMS world, we're talking in microns here. So these failure mechanisms aren't understood very well, but they're very important to the scientific research that we're doing here. Mentioned earlier, uncertainty. And when you think about anything that you're doing in testing, there's always inputs. There's, in a simulation model, there's inputs, there's a simulation model, there's outputs. And one of the things that when we make our assumptions, we have to define what those uncertainty areas are. So whenever we're doing these, the simulation endpoints are inherently uncertain. There is always variability. There are ranges of operating conditions. So those are the things we're trying to capture when we're incorporating the concept of UQ into these simulation models. Very simply, as you can see from the graphic, questionable inputs, I shouldn't say questionable, but uncertainty in the inputs go into the model. There's, of course, has to be uncertainty in the outputs. And that's what we're utilizing and why the cyber infrastructure is so important to the success of this program. And remember what we said, that the device is that front view of what the device looks like. This is an image of the switch not actuated, and this is an image of the switch with the membrane depressed, and basically the switch is activated. As you can see, in this example, this isn't an exact number. There's some uncertainty involved there. That's where the uncertainty quantification comes into. We talked about the device scale. This device is at the micron level. So at the micron level, we're talking things like electrostatics, charge transport, and all those other components, those standard components of electromechanical devices at that level. But we're also incorporating at the micro scale. We're talking nanometers when we're talking about the actual space where the dielectric and the membrane is touching the dielectric. This is in terms of nanometers. This is very small. The contact area, what happens to that contact area after millions, billions of cycles? How does the surface chemistry change over time? These are the types of things that we're incorporating into our models. Keep in mind that, once again, this is, billions of cycles. So these are types of areas that were not, as I said, not relatively well understood at these scales. So these are what we're incorporating into our model. I said at the beginning that we're also incorporating an experimental program, because one of the components of this is we're trying to experimentally verify and validate some of the results that we're getting from our simulation model. Anywhere from things like the microstructure characterization of the materials, talking about the dielectric charging, where there's lots of models that are currently out there that are doing these types of calculations. We are doing some things from vibration analysis in a mechanical, specifically in a true laboratory sense, and other things trying to quantify the uncertainty in other known devices that are out there. We're taking these types of results and trying to verify and validate against our simulation models. Here's the architecture of the Prism software that we are utilizing. It's a tool that we're calling Mimosa. And Mimosa has within it models and solvers from different components of code. There's about, I think, like 15 or 16 different components into this model, into this overall framework that are being used. Anywhere from the setup of the input data from ANSYS or Qubit or Pro-E to the LAMPS software that we're utilizing at the national labs, to other types of visualization software like ImperaView. So the whole framework is utilizing a lot of different components, and these components are being run across different community clusters here at Purdue and also on the national labs. The ones that specifically we're using at this time, Prism was one of the purchasers, if you remember from John's presentation earlier, a member of one of the community clusters. So we're utilizing Steel and Coates as well, and Lawrence Livermore and Los Alamos supercomputers for some of the simulations that we're doing. I know I flew through this kind of quickly, but why Prism and what is it about? Prism is put into place because it's a very interdisciplinary program. We're using faculty from aeronautics, from ECE to ME. We have partnerships with the Berkeley Nanotechnology Center, which is where we're doing a lot of our experimental testing. Sandia National Labs, access to some of the National Lab supercomputers. Impact, the center over at the University of Illinois. In that center, we're utilizing some of the codes that we are developing are available and accessible on NanoHub, as well as on MEMS hubs as well, which is another aspect. It's another one of the hubs here on campus. And simply put, we have some really good people here at Purdue. So students in the top-ranked programs across the country. I know it was kind of fast, but we're just another example of why we're using cyber infrastructure here at Purdue. That is all I have. I want to turn it back over to Pete, and we can go to questions. All right.
Go ahead. See if David and Gabriela will be able to come back up. We'll keep this rather informal, but are there any questions from the audience for the three speakers today? Yes. Wherever you prefer.
Yes, I had a question for Gabriela. With regard to the use of instruments by undergraduate students, you mentioned a few instruments. I wanted to also raise thermal analysis and calorimetry as well. But in many cases, the real art and the science comes in specimen preparation and specimen mounting, not so much in the use of the instrument. And how do you deal with those issues?
So the types of instruments that we have put on the network at the moment are those where the samples actually can be prepared by a group of students and shipped, and then basically just put into the auto sampler sample holders. Or in some cases, they're put into the auto sample holders on location, and the entire sample holder is shipped with those samples already in it. There are some instruments that really aren't going to lend themselves to this approach. So not every instrument would be a viable candidate for doing this. In some cases because of the reasons that you alluded to, but in other cases because there may not be an auto sampling capability for the types of measurements that a particular instrument is making. And we've already run into that with some of the experiments that called for particular types of instrumentation that we just couldn't put those instruments on the network. Now, that said, some of the sample handling situations are dealt with by having an expert instrumentation manager on site. So these instruments are not, sitting there being handled on location by undergraduates or by graduate students. We have a staff member, and in some cases more than one staff member, whose job it is to handle the samples as they arrive and then put them into the right containers and deal with the network and all of those sorts of things. The conversations that we're having with folks out at Burke and Bindley involve instruments where there is a single person that is actually associated with those instruments and would handle those types of situations where the expertise is needed to actually mount samples.
Thank you. Do we have other questions? Yes.
Would you say a few words about the experiments that you are replacing by having these instruments online?
Is that for me?
Yes.
Okay. Are you talking about the experiments that are part of the curriculum normally? I can say many words about that depending on what angle you want me to come at it from. The approach that we developed for teaching undergraduate courses this way was designed to be adaptable to the specific situation at whatever institution is implementing it. So as I mentioned, we have 17 institutions nationwide. So the instructors at those institutions have the decision over which experiments they want to replace in their curriculum with these research experiments. When we look at the curriculum and the ways that we have designed the research experiments to be implemented, we have taken basic techniques that students are normally introduced to in these experiments. You know, in these courses in first and second years, often what happens is you have a technique or a procedure that a student learns in an experiment, and then the next week they learn something totally different that really isn't related to that, but it's another technique. In these research experiences, the students are in fact learning many of those same techniques, but they're applied to this overall research question. And so if the goal of the instructor is to make sure that students are introduced to the basics of an acid-base titration, or you know, some other technique that they need, you know, to use a spectrometer, for example, then those procedures will still be part of the student's curriculum, but they'll be integrated with a learning experience that allows the student to see how techniques and hypothesis formation and the use of evidence to make scientific claims are all an integrated part of the science endeavor, rather than having it be a series of individual technique learning experiences. So we feel that the laboratory curriculum is still meeting its learning objectives through this integrated research approach, even though 6 experiments or 8 experiments are being replaced by this one long-term experience.
Thank you. Any more questions?
I'll ask a second one. and I'm still focused on Gabriela, although I would like to think of something to ask David also. I'm reminded of a National Research Council report having to do with forensics laboratories, and the problem with expectations that because of CSI, forensics laboratories can do all the things that you see on CSI, which they can't because they can't afford the instrumentation. It seems to me that the capability you've built up would be ideal for a regional resource that could service forensics laboratories in pretty much an automated way in order to develop evidence. And what thoughts do you have on that?
Well, as I mentioned, and I see Al in the back grinning, because the conversations that we've initiated with the other Discovery Park centers are along those lines. The idea is that you know, we may be able to be the center of a regional resource that would provide laboratories with industrial or other research types of needs, where they may not have access to all of the instrumentations, all the instruments that they need on site, or where a consortium of users knows that they all would like to have access to an instrument, but there's no real need to buy more than one, or perhaps it's just too expensive of a resource to have more than one. So long term, that is the kind of approach that we would like to see this technology applied to, some sort of a regional resource.
I could actually add a little comment there, seeing as I want to have a go too. So actually, as part of the project that I discussed, and I didn't have time to mention it, but actually as a component of our project, we also have an outreach piece to actually take samples from the community and run them on our instruments, and then deliver the data back through the cyber infrastructure. And so I think this model is certainly applicable to those situations where an instrument's too expensive for other labs to have, for example. But I think there are some significant issues to be dealt with in terms of metadata tracking for the samples, you know, making sure that you have the data handling systems in place so that you don't get the samples mixed up, and the labeling issues. I mean, these are trivial, but as you know, it's super critical. Exactly, but I think that the concept is definitely great.
How do you do the cost sharing on the instruments? I mean, if one institution uses more than another?
Well, that's an interesting question because right now the costs are being borne by the NSF grant that we got to establish all of this, and we are looking at how, in fact, we would do billing, especially, you know, this is a system that was originally developed for educational applications, as I mentioned, and when we do the math and figure out what the costs would be, for example, to a 2-year college, they're really much higher than any 2-year college would likely be able to afford. So part of the reason for branching out to include industrial and research partners is potentially to be able to support some of the educational costs through some of the partnerships that involve industry.
So I have a question for Dr. Salt. You've amassed this database of information that is available to others. Is there any mechanism for the community or other users to give back, to add to your repository? And if so, you know, what kind of checking is, you know, can anyone just upload data, and what kind of checking is made to make sure it's valid?
Yeah, that's a great question. So currently the only people who can input raw data to the system would be people who are direct collaborators of the people who run the project, so myself and my other collaborators. So that currently happens, so those people are kind of pre-vetted, if you like, right? But I think maybe what's perhaps also very, very critical for this kind of endeavor is the idea that the larger research community can add back annotation. So, you know, to give a concrete example, let's say we take an organism which we know one gene has been knocked out, we run it through our system, we see that it has these particular characteristics. We present those characteristics in our database, but we're not focusing on that gene. We don't really understand what that gene does. Maybe you're an expert in that gene and you come to our database and you're very excited that when you knock it out, it gives this phenotype. What we would like to be able to do is for you to be able to come in and annotate that data with your expert knowledge and say, hey, well, I know from my experiments this gene does this and this, and so you can build up knowledge in the database. And so I think that actually those kinds of tools are what we're going to be adding in with this new development that we're going to be funded from the National Science Foundation. And I think different models have been tried to kind of to curate that kind of annotation. And I think probably the best way is a community type of policing, if you like. So that one model is that you have individuals who become kind of editors on particular gene sets, for example. And so if you're an editor on your particular gene family in this ionomics community, then when someone annotates a gene that's in your family, then you would get a little instant message saying so-and-so has annotated this gene. Do you approve that as being kosher? And you would say yes or no. So I mean, it's a great question. I think that's definitely the future of these large-scale functional genomics projects. But we need to develop the community tools that are going to let those kinds of activities go forward.
I did think of a question to ask you, David. I'm interested in the interrelationship of the plant and the soil, and what you're learning through your ionomics that answer questions like what nutrients are dependent on other nutrients, such as nitrogen and carbon, and what would lead to better utilization of natural fertilizers rather than artificial fertilizers?
Yeah, so that gets at the heart of one of the things I'm extremely interested in, which is the mechanism of local adaptation in plants. And so, for example, if you know, we're all very familiar that the population, you see different kinds of flora in different kinds of environments. And so one of the things that we're trying to understand is what are the genes and gene networks that are driving that adaptation. And so I briefly touched on one slide where I showed that we're developing this tool which lets us map different genotypes across the landscape. And then overlay different environmental conditions, and soil properties are one of those. And so the idea would be that we might, and we've already just recently published an example of this, where you see a non-random distribution of a particular type of natural genetic change, and that's highly correlated with a particular distribution of, let's say, a soil nutrient. And so that would then become the first piece of evidence which would suggest that genetic change is adapting that plant to that particular altered soil environment. We can then go back and set up a new set of experiments, kind of iterate that whole process around, see if that's a correct assumption, and then we can then actually extend our experiments out into the field, where we could actually go and test then in the field whether that gene is really adaptive. And then the idea would be that with that understanding, you could then bring that back into crop systems and try and improve crop systems to grow under those particular sets of conditions. So that's one of the big areas that we're interested in.